angles, what are they again? Phi and psi, okay? That have, they're restrictive in the angles that we can have. Why? Because what are they flank? What are they on the other side of? The peptide bond. What do you know about the peptide bond? Planar, okay? And it's planar because it, even though it looks like a single bond, it's planar because it has resonance. All right, it's a partial double bond pair there, okay? And so that keeps things in very specific types of conformations then, okay? So there's certain what we call allowed phi and psi angles, okay? And that's what we see with these certain secondary structures. Quick review on the alpha helix, right? Do we have hydrogen bonds within the helix? Between what residues? 1 and 4, okay? Those are where we have those. How many residues per turn then? 3.6, all right. 3.6 residues per turn, and then you have a hydrogen bond interaction. Where do the R groups point on an alpha helix? Out. Why? What do we know about the middle of an alpha helix? Very, very easy. What R group could maybe fit in there? There we go. All right, beta sheets. Where do the R groups point in beta sheet? In and out of the plane, right? And they actually, do they all go out? Or they alternate in, out, in, out, in, out. So if you get an amphipathic alpha helix, or excuse me, an amphipathic beta sheet, in the primary structure you could see potential every other one repeats with the same type. Okay? Questions for me? Then we talked about random coils, but basically random coils, everything that's not alpha helix or beta sheet. Okay? Does that mean random coils are less important? Questions for me from last time? Yeah. Right.
So if in beta we see an alternating every other, in alpha do we kind of sometimes see an alternating every 4, like maybe?
Yes, and that's exactly what you should see, right? That's something we're going to talk a little bit more today, but that's also, we didn't technically go into it, but it's on the homework about an amphipathic alpha helix. So every 1 and every 4 should be similar, whereas 2 and 3 should be also similar because they're on each side. Everybody got that? Remember in the homework? There was drawn amphipathic alpha helix. I don't care how you draw it, you can simply draw this. You would say if this is A, B, C, D, A and D probably are what? Let's give it an example. Polar, okay? That would make B and C if you wanted to make this amphipathic, meaning half water loving and half water hating. B and C would have to be non-polar, so you would divide this down the middle. Everybody okay with that? Which side of that sits near the inside of a protein probably? More than likely. The non-polar phase. Okay. So in an alpha helix, if you see those repeats every 4, that's a good indication. Is it a guarantee that you have an alpha helix? No, but something you can maybe look for. Yeah, good question. Did that answer your question, Ryan? Other questions for me?
So you said beta sheet, the R groups either it forms all again or
outer. Basically, you have the peptide bond, and if you have the peptide bond considering this plane, the beta sheets point in and out of that plane. Yeah. So if you think of like a tetrahedron, right? Remember when you guys did tetrahedrons and tetrahedral geometry in Vesper? Yes, yes, yeah. Remember you had 2 things in the plane, you had 1 thing in and 1 thing out? Same kind of thing. One of those things that's a tetrahedron is what the C alpha is always a tetrahedron. The R group can point out, and then the H goes back, and then the next one, the R group goes out, goes in the back, H goes out. Does that answer your question? Other questions? Okay, so now let's talk about different types of proteins. Now, when I say different types of proteins, we're going to be very, very generous with the grouping that we have here, because if you wanted to, you could get very, very specific with types of proteins. We're going to talk about structural characterization of proteins. There's 2 main types, okay? It's classified more by their structure and then therefore their function and their extension. The ones that we're going to talk about are number 1, fibrous proteins. Okay? Fibrous proteins are more like fiber pieces, right? They're very strong, they're very elongated. Okay? What's nice about fibrous proteins is they're very easy to spot because they usually have a repetitive primary sequence. If you see a primary sequence that has like 7 or 8 residues that just constantly repeat, is that a guarantee you're going to have a fibrous protein? But is it a good indication you might? Yes. Okay? So we'll talk about 2 examples. Alpha keratin, you may have heard of these, and collagen. Okay, those are 2 examples. And then we'll talk about globular proteins. Basically, globular proteins is every other protein that's not a fibrous protein. Okay? there's lots of different types of globular proteins. Okay? But the main thing is that they have very different structures. Okay? Because they have very different functions. Okay? So, we're going to talk more about hemoglobin and myoglobin. Okay? Which, again, we've already seen hemoglobin in the lab a little bit, but we're going to talk about why it has the color and what its structure looks like. It's a type of globular protein. It has an irregular sequence. So it's usually, and this is not always the case, but it's usually harder to predict globular structures. Why? Do you think they have repetitive sequences? Not really. Okay. Is that a guarantee that it's always going to be easier to predict a globular structure? Probably not, but for the most part, that's generally what we see. Okay. Questions from two different types of proteins, fibrous and globular. Okay. All right, so here we go with fibrous proteins. So, on the right here, what we have is this is an alpha helical, what we call alpha helical wheel. Okay, so you notice on the board here, I drew 4 residues, A, B, C, D. Same thing, this is basically what this is drawn, except taking this, flipping it out towards you 90 degrees. Okay, so this is looking down the barrel of an alpha helix. So A would be our first residue, A, B, C, D. Remember, 3.6 residues per turn, 1 to 4. Okay, which means my A and D residues, what do you notice? They probably have what? Similar characteristics, whereas B and C out here probably have also similar characteristics. Now, if you look from D, we go down to E, F, and G. That's a complete turn, okay, around an alpha helix. All right. So this is what we're talking when we talk about fibrous proteins. Okay. An easy way that you can tell if you have a fibrous protein is they tend to have a repetitive primary structure. Okay. Now that depends upon what secondary structure dominates. Okay. In this case, our secondary structure that dominates is an alpha helix. So you should see repeats every 1 in 4 should be similar. Okay? And what this does is if you have those same repeats over and over again, you have the same primary structure, which means you're probably going to get the exact same secondary structure. Okay? And the more of that secondary structure you have, just repeated, basically the stronger protein that you're going to get. There's no variability, there's no pieces that are kind of floppy. Everything's stiff, everything's aligned just right. Okay? So you get something that is very stiff, okay, and very compact. And that speaks to the function of these proteins. These proteins are not here to move around and be floppy, they're there to produce some sort of rigidity. Okay? Anybody heard about, like, you've probably heard about collagen in the news, right? Or on TV or something like this. When we think about collagen, we think about what? Injections, right? Yeah, because it's a protein that basically plumps things up and is stiff and holds things in place. Okay? It holds like your skin in place, you don't get wrinkles and things like this. Again, this is always usually, in biochemistry you have to qualify a lot of things by usually. It's usually not an always thing. Usually in fibrous proteins, there's only one main secondary structure. Okay? So if you were to look here at this example, what type of secondary structure is this 100%? Alpha helix. You notice there's really no random coil in there, it's just all alpha helix all the way throughout. Okay? Now does that mean that you can still have a fibrous protein that maybe has a little bit of beta sheet or a little bit of random coil? Sure. Usually it's dominated by one type of secondary structure. Questions for me? Okay, so again, we know structure dictates function. Okay? So that means when we look at these fibrous proteins, they're very stiff. They're elongated. Basically what they do is they're there in some sort of structural role. They keep shape and they keep integrity of certain things. They keep integrity of your cells. They make sure that they're not, what's the word when they fall in on each other? When they're falling in. Anyway, what? Collapse. 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 There's another word. Anyway, doesn't matter. So they help with cell shape. They make sure your cells stay, you know, or whatever, fill in whatever you want there, but it basically helps maintain integrity. Okay? Main thing, again, structure dictates function. Okay? So that's what we're looking for. If you see something that's stiff, something that's elongated, doesn't really have a lot of flexibility, you're not going to see it actually be really flexible and have a lot of movement. It's going to be there to make sure other things stay in line. Okay? And that there's good integrity for anything, any other protein, so that they can move around, they have space, whatever it is. Okay? Questions for you? we're going to take a look at something called alpha keratin. What do you think secondary structure dominates alpha keratin?
Beta sheets.
There's also some. So alpha keratin is dominated by alpha helix. Guess what beta keratin is made up of? It's crazy, I know. Biochemistry names are really tough to remember. So, alpha keratin. Alpha keratin, we're going to talk about ones in mammals. Other different species have different versions of alpha keratin, but we're going to talk mostly about ones that are mammals, okay? This is a fibrous protein. It has what we call a coiled-coil structure, okay? That's what this is right down here on B, okay? What it is it's two separate polypeptide chains that are both coils that coil around one another. Coiled-coil, we call it. Kind of like, am I updating myself? I say, remember landline phones with the long cords? Yeah. Okay. Remember those really long telephone cords you had and the coil would actually coil around itself? That's a coiled coil. That's an example of it, okay? Now, if you had that telephone cord, just one single coil, it's pretty easy to move around with, right? How easy is it to move around if it's coiled coil around itself? Not. So is it stronger or weaker when it's a coiled coil, two things around itself? Stronger, okay? So we're looking here at a stronger protein. So again, when we deal with fibrous proteins, all of their primary, secondary, tertiary, quaternary structures have to deal with making the protein stronger or somehow making its function, okay, more appropriate, right? If you wanted a protein that's there for stiffness, that's there for structural integrity, and you gave it quaternary structure that made it more floppy, that defeats the purpose, okay? In this case, the coil gives it more rigidity, okay? So again, the two helices, they actually wind around one another. And what they actually do is they actually wind around each other in a left-handed conjugation. It's left-handed because, if you remember, alpha helices are always themselves right-handed. By taking and non-natively folding these things around the other way, it confers more structural rigidity, confers more stability, okay? Because it's not necessarily, we always see things going to the right. If you put it to the left, it's going to take a really unique protein, a really unique enzyme, a lot more energy to unwind it because it's not usually what we see. But it gives it more stability that way. Makes it more compact. And the pitch of the coiled coil, okay, so two coils winding around one another, normally if you see that with a regular alpha helix, it's about a 5.4 pitch where you go from one piece to the next. Okay. Here in alpha keratin, it's actually less than that, so it's more stiff. You're making kind of like the springs, like in physics, right? I always talk, oh yeah, you have this frictionless spring that does no gravity. Yeah, it exists. Anyway, so you have, when you have an alpha helix, you have something like this. But if you make it more compact, right, you make it what, more or less stable? More stable, okay? Now, you don't want to make it too compact because you need space for the amino acids that sit in there. But the more compact you can make it, the more stable and stiff that you can make it, okay? Primarily, we see alpha keratin in things that are relatively stiff in our bodies, in your nails, in your hair, things like this, okay? Questions from me on this? We're going to talk now about the 4 levels of protein structure of alpha keratin.
So the pitch is how tightly it's wound then?
Yep, relative to each other.
Okay.
Yep, so these two coils wind around each other. It's not 3.6 because then you'd have every single coil itself hitting every coil. So it's around, think about that telephone cord, each of it going around and around. Does that answer your question?
So it's not how tight they're individually coiled, it's how
right, it's how tight these two
okay. All right.
separate polypeptides are coiled around each other, yeah.
Right.
The individual ones are still the 3.6 residues per turn, same normal alpha helix, yep. Yeah. Other questions for me?
So like right-handed versus left-handed, you're saying, like if looking down, it's going...
Right, well if you look at the individual alpha helices, they still go right-handed. But when you look at the two, they're wound around each other, can't say that, in a left-handed conformation. It's hard to see without a model. And I'll bring a model next week, or not next week, on Wednesday, if you want to see that, but it's easier to see it in 3D rather than me drawing it on a two-dimensional board. Other questions for me? Okay, so here we go. Now let's talk about